Over 540 million years ago, the Earth's oceans were dark, sediment-heavy, and entirely devoid of complex life. Biologists have spent decades trying to identify the exact species that broke this pattern, the very first animal to exist in this primeval world. A team of geochemists at MIT managed to trace this lineage, but they didn't do it by looking for fossilized bones. Instead, they extracted samples of 541 million-year-old sediment to search for invisible chemical fossils, remnants of biomolecules preserved in the rock for eons. This diagram shows the chemical fossils they found, steranes with 30 and 31 carbon atoms. These are stable sterols, like cholesterol found in cell membranes. Manufacturing them requires a distinctive enzyme produced by specific genes, a process impossible to replicate by random geological chemistry. The genetic code required to produce these exact molecules traces back to the sea sponge. Our earliest animal ancestors survived without a brain, a heart, or a nervous system. Early naturalists classified sponges as plants because they appeared to be purely stationary and lack any recognizable internal organs. Looking at these vintage biological illustrations, the floral silhouettes made them nearly indistinguishable from underwater flora. To find the true boundary of the animal kingdom, we have to look past the lack of a mouth or muscles. Modern biology defines animals by multicellularity, the ability for distinct cells to adhere, communicate, and specialize to perform mutually dependent tasks. Molecular testing confirms that the sponge was the first organism to cross this threshold, making it more closely related to a human than a cactus. Understanding the sponge reveals the foundational biological blueprint that eventually allowed for the development of all complex animal life. A sponge operates as a decentralized machine, relying entirely on isolated, specialized workforces rather than a central command center. It feeds without a stomach or digestive tract. Instead, every cell is responsible for its own intake through intracellular digestion, breaking down food particles that must be smaller than the individual cells themselves. This cross-section shows the choanocytes, or collar cells, that line the internal chambers. Their tail-like flagella beat in unison to act as a microscopic water pump. This motion draws water through the outer pores, filtering out bacteria and plankton to be ingested by phagocytosis. The sponge's most powerful tool is the archaeocyte. These are totipotent cells, meaning they possess the flexibility to transform into any other cell type the sponge needs. This cellular plasticity allows a sponge to constantly repair and regenerate. If a section of the wall is torn, archaeocytes migrate to the site and transform into the specific structural cells required to seal the gap. Biological decentralization allows the sponge to survive conditions that would be fatal to more complex organisms, such as being shredded or pulverized, as the individual cells simply reorganize into a new individual. But a creature made mostly of moving cells and a jelly-like middle layer still needs a way to maintain its shape against the pressure of ocean currents. This is a microscopic view of spicules, rigid skeletal structures manufactured by the sponge from silica or calcium carbonate. These geometric rods and star-shaped spikes provide the internal tension necessary to hold a form. Because these spicules grow in varied shapes like monaxons or tetraxons, they interlock to create a three-dimensional architectural scaffolding. This mesh supports the soft tissue while allowing water to flow through the body. This microscopic scaffolding scales up into macrostructures like the Venus flower basket, proving that resilient structural engineering doesn't require a brain to design it. This modular biology has allowed sponges to outlast multiple mass extinctions and endure extreme climate shifts for over 500 million years. To survive severe droughts or freezing, freshwater sponges pack their totipotent archaeocytes into armored pods called gemmules. These protective coatings of spongin and spicules allow the cells to remain dormant until favorable conditions return. In the nutrient-poor deep Pacific, some species have abandoned filter feeding entirely. The harp sponge has evolved into a carnivorous predator, using hook-like structures to snare passing shrimp. By foregoing a fixed, rigid anatomy, the sponge can morph its biology to conquer almost any ecological niche, from coral reefs to the deep abyss. Billions of sponges, acting as the world's first filter feeders, changed the very chemistry of the primordial oceans. A single sponge can filter many times its own volume in water every day. This relentless activity removed massive amounts of suspended organic debris, clearing the murky Ediacaran seas and increasing the depth at which light could reach the seafloor. This filtration shifted the flow of energy in the ocean. Sponges captured dissolved nutrients and bacteria from the water column and concentrated them into their own bodies, which then became a food source for larger, more complex invertebrates. By clarifying the water and stabilizing the nutrient cycle, sponges constructed the environmental conditions necessary for the evolution of all subsequent animal life. Because sponges are sessile and cannot flee from predators, they spent 500 million years perfecting a massive arsenal of chemical defenses to deter fouling organisms and competitors. Modern medicine now relies on these 500-million-year-old bioactive compounds. They are currently used to develop new antibacterial, antiviral, and anti-cancer treatments, turning ancient survival strategies into human cures. The origin of the animal kingdom traces back to a simple, powerful discovery: individual cells learning how to specialize and work together, a biological collaboration that still sustains every human organ today.